In this lecture, we're going to learn about the different neurochemicals that exist within the nervous system, and then we'll also end talking about the ways in which drugs, either of abuse or medicinal value, can modify neurotransmitter function in the nervous system. If you're like me, you probably start your day off with some sort of caffeine. My caffeine of choice is a cup of coffee in the morning, sometimes two. Some people use caffeine all throughout the day. I'm not one of them. I can't fall asleep at night. However, I will say, and most of my family would agree, that I am addicted to caffeine. And so one of the things we'll talk about is the ways in which caffeine not only, might avoid the withdrawal symptoms for not consuming it if you're addicted like me, but also the ways in which caffeine stimulates the nervous system to increase things like attention, arousal, and focus. But we're going to be discussing neurotransmitters in general terms. First, we'll talk about individual neurotransmitters, focusing largely on classic neurotransmitters, and briefly touching on neuropeptides, just because there's hundreds of them, and they don't get as much attention as the classic neurotransmitters. And then we'll dive into the ways in which drugs, including caffeine, can modify neuronal function and ultimately affect behavior. So this is going to be the outline of today's lecture. We'll talk about neurotransmitters, the different kinds of classic neurotransmitters, aka small neurotransmitters that exist in the nervous system. And then we'll briefly touch on, by brief I mean like one slide, on neuropeptides, because they largely serve to modulate or modify the actions of the classic neurotransmitters. The classic neurotransmitters are the ones that are doing most of the work in the central nervous system. Then we'll talk about the pharmacology of synaptic transmission, what the 6 steps in neurotransmitter action are, and how drugs can affect that neurotransmitter action. And then we'll briefly touch on categories of psychoactive drugs. These are drugs that have an action within the nervous system and produce psychological or behavioral changes. We'll briefly touch on drugs that increase and drugs that decrease behavior. However, if you're interested in this part, or this lecture in general, consider taking neuropharmacology, which is a course I often teach, and it's a capstone course in the program. Okay, so this is a chart from your textbook that talks about the major categories of neurotransmitters. Your textbook categorizes monoamines, amino acids, peptides, and then it groups acetylcholine, which is one of what I consider a classic neurotransmitter, in the other because it doesn't necessarily fit into a category. So it's a category in and of itself because it doesn't kind of contain characteristics that are similar to other neurotransmitters. So we're going to be focusing on 1, 2, briefly 3, and then acetylcholine as well. So let's get started. So I've broken up the neurotransmitters that we're going to be talking about into kind of this chart, and we'll go back to this chart as we change between categories. We'll first talk about amino acids, and there are 2 major neurotransmitters within the amino acid category. The reason why they're called amino acid neurotransmitters is because the molecule that's used to make them are amino acids, or in fact they are amino acids, but they're slightly modified so that they can serve as neurotransmitters or chemical messengers. We'll first talk about amino acids. So the 2 amino acids which we get the precursors from our diet for glutamate and GABA. Glutamate and GABA are really important to talk about because they are, one, the major neurotransmitters in the brain, right? Most of the functions of neurons occurs through one of these 2 neurotransmitters, and they also oppose each other in their action. Glutamate is the major excitatory amino acid neurotransmitter, so every time it gets released at the synapse, it produces an EPSP response in the postsynaptic cell. GABA, every time it gets released, it is the major inhibitory amino acid neurotransmitter, and so when it gets released, it always produces an IPSP, or an inhibitory response, on the postsynaptic cell. And as I referred to, but pretty much all communication between neurons in the central nervous system, almost all of them, are accomplished by one of these two neurotransmitters, and then the other neurotransmitters may be used, but more selectively. So the vast majority of neurons are utilizing glutamate and GABA to communicate within the nervous system to perform its functions. Let's talk about glutamate first. Glutamate is synthesized from the precursor glutamine. An enzyme takes glutamine and converts it into the active neurotransmitter glutamate. So glutamine is the precursor, glutamate is the neurotransmitter, and the enzyme that makes glutamate is called glutaminase. Once glutamate is released at the synapse, it can activate multiple different receptors. So we talked about neurotransmitters having classes of receptors, and a single class is referred to as a subtype. So glutamate has 4 subtypes of receptors. All of them ultimately produce EPSPs in the postsynaptic cell. Glutamate can bind to and activate 2 different types of ionotropic receptors, AMPA and kainate. They're given these names because of the drug that can activate these receptors, and we know that the receptors are separate because these drugs selectively activate these receptors. The other ionotropic receptor is NMDA. It kind of sits in its own category because glutamate alone does not fully activate NMDA receptors. It requires depolarization of the postsynaptic cell to fully activate NMDA receptors. And so that's why it sits in its own category, separate from AMPA and kainate receptors. But all 3 of these are ionotropic receptors, meaning that they are channels that get activated or open when glutamate binds to them. NMDA receptors are also really important, have been well studied in the process of learning and memory. The last subtype of glutamate receptors is the metabotropic glutamate receptor. Just like metabotropic receptors, this mGlu-R, which is what it's typically known as, has a G protein, so it is not an ion channel, but its actions following activation results in EPSPs in the postsynaptic cell. GABA is, interestingly enough, synthesized from glutamate. So it's ironic, or I guess interesting, to consider that the major inhibitory neurotransmitter is synthesized from the major excitatory neurotransmitter. So the precursor for GABA is glutamate. The enzyme is known as GAD, glutamic acid decarboxylase. And then GAD takes glutamate and makes GABA from it. So the precursor is glutamate, the enzyme is GAD, and the neurotransmitter is GABA. All receptors of GABA, because it's the major inhibitory neurotransmitter, once activated produce IPSPs, and there are 2 major subtypes of GABA receptors. We have GABA A receptors, which are ionotropic, and once activated, they always produce IPSPs, which inhibit neuronal function, making action potentials less likely. Drugs like alcohol and benzodiazepines produce their effects by binding to GABA A receptors. The other subtype of GABA is GABA B. It is a metabotropic receptor, so it has a G protein attached to it, but it too, once activated, ultimately produces IPSP, or inhibitory responses, on the postsynaptic cell. So moving on to monoamines, we have 2 types of monoamines, catecholamines and indolamines. We'll first talk about catecholamines and then we'll discuss indolamines. So there are 3 types of catecholamines that you may have, some of which you may have heard of: dopamine, epinephrine, and norepinephrine. Monoamines are called monoamines because they contain a chemical structure called amine, and they only have a single amine. So monoamines are called monoamines because they all contain a single amine group in their chemical structure. They're also all made from amino acids, but they're not considered amino acid neurotransmitters because they are different in their structure. So all catecholamines are made in kind of the same pathway. So remember, catecholamines include dopamine, norepinephrine, and epinephrine. The precursor for all catecholamines is the amino acid tyrosine, which we get from our diet. The enzyme tyrosine hydroxylase takes tyrosine and makes it into L-dopa. Another enzyme, which you don't need to worry about, takes L-dopa and makes dopamine. If a neuron expresses enzyme 1 and tyrosine hydroxylase, and that's it will release dopamine as its neurotransmitter. However, if that enzyme also makes enzyme 2, it's going to take that dopamine and make norepinephrine from it. If a neuron has tyrosine hydroxylase, enzyme 1, enzyme 2, and that's it will release norepinephrine as its neurotransmitter. However, if it also makes enzyme 3, norepinephrine will be converted to epinephrine by enzyme 3. So which neurotransmitter, which catecholamine the neuron makes is dictated by which enzymes are present in the neuron. Okay? So the expression of the enzyme in a neuron dictates which neurotransmitter it will be making. If you're not familiar with it, epinephrine is also the same exact chemical as adrenaline. Norepinephrine is the same exact chemical as noradrenaline. But typically, adrenaline and noradrenaline are seen as hormones released by glands, whereas epinephrine and norepinephrine, despite being the same exact chemical, are typically found in the nervous system. So let's talk about how these neurotransmitters are released and removed from the synaptic cleft. So we're going to be using dopamine as the example, but it applies to all catecholamines, not just dopamine, right? So in this example, we're using dopamine, but what I'm going to be sharing in the slide applies to norepinephrine and epinephrine as well. So like classic neurotransmitters, dopamine and catecholamines are synthesized in the presynaptic terminal or the terminal button. It gets packaged into vesicles and released through the process of exocytosis. It's going to bind to and activate dopamine receptors, which produces a response in the postsynaptic cell. After it pops off the receptors, it gets removed from the synaptic cleft by reuptake transporters. Once in the terminal, it can be broken down by an enzyme called monoamine oxidase, or MAO, into molecules that are no longer active. However, dopamine can also be repackaged and reused for re-release. This entire process also occurs for norepinephrine and epinephrine. The same enzyme, a different reuptake transporter, and different receptors are present, but the process is the same for norepinephrine and epinephrine. So let's talk about the receptors for these catecholamines, starting with dopamine first. Dopamine has 5 different subtypes of dopamine receptors, all of which are metabotropic, which means they use G proteins and second messengers. I'd mentioned this in a previous lecture, but the dopamine receptors, the 5 subtypes are D1, D2, D3, D4, and D5. Not very creative in naming the receptors, but that's what they are. They're all subtypes of the dopamine receptor, and they're all metabotropic. Dopamine is known to be involved in a variety of different functions depending on where in the brain it is and what receptors are being utilized. It's known to be involved in movement, and Parkinson's disease is caused by dopamine dysfunction. It involves in attention, learning, as well as the reinforcing effects of drugs in the reward pathway, which we'll talk about in another lecture. So here is what's really interesting. Dopamine, decrease in dopamine activity is what results in Parkinson's disease. So the way they treat Parkinson's disease is to provide the precursor for dopamine, which is L-DOPA. If you remember from the synthesis of catecholamines, L-DOPA occurs just before dopamine is made. And we administer L-DOPA to patients with Parkinson's disease, at least in the earlier stages, because the L-DOPA can cross the blood-brain barrier, whereas dopamine can't. Interestingly enough, it is thought, although not entirely, I guess, confirmed or validated or consistent, that increases in dopamine activity contributes to psychosis symptoms in schizophrenia. And the way they treat schizophrenia is by blocking specific types of dopamine receptors. What's interesting is that a side effect of too much L-DOPA can produce psychotic-like symptoms, so symptoms that are similar to what we might see in schizophrenia, but it's purely caused by L-DOPA treatment. And a side effect of antipsychotics, which is what the category of drugs used to treat schizophrenia is called, can produce Parkinson's-like symptoms. It's not Parkinson's disease, but caused entirely by the actions of antipsychotics. Norepinephrine and epinephrine both utilize the same group of receptors, and they're called adrenergic receptors. They both bind to the same category of adrenergic receptors, all of which are metabotropic, but they perform different functions in the nervous system. Norepinephrine appears to be important for attention and arousal, as well as feeding behavior. It participates in sleeping and dreaming. Epinephrine in the nervous system, but known as adrenaline in the periphery, or like the rest of your body as a hormone, may help to control things like eating, as well as functions in your visceral, like your motor function. All right, we've talked about catecholamines. It's now time to talk about the other monoamine, indolamines. And while there are other indolamines, we're going to focus largely on serotonin. Serotonin, you likely are familiar with if you've taken any psychology course. Serotonin is an important neurotransmitter for a variety of different functions. Like other monoamines, serotonin has a single amine group in its chemical structure, and it is produced like catecholamines from an amino acid, which we get from our diet. This amino acid is known as tryptophan. An enzyme takes this tryptophan and makes 5-HTP, or 5-hydroxytryptophan. So you can abbreviate this molecule as 5-HTP. A different enzyme takes 5-HTP and converts it into 5-HT, 5-hydroxytryptamine, which is serotonin. You'll notice that sometimes I'm going to abbreviate serotonin as 5-HT, denoting its chemical structure, but both 5-HT and the word serotonin refer to the same neurotransmitter. Serotonin does a whole bunch of different things, but you may be most familiar with its role in mood, which seems to be the underlying cause for depression, although that's not definitive. It has been known to play a role in emotional behavior as well as sleep, but also eating and attention. So serotonin does a ton of different things in the nervous system. Serotonin transporters, right? So all monoamines are removed similarly as dopamine in the prior example that I told you, and we'll talk about serotonin in particular in a second. But the way some antidepressants work, not all, but some antidepressants work to block serotonin transporters to keep serotonin in the synaptic cleft longer. So there are selective serotonin reuptake inhibitors, such as Prozac, you may have heard of, or Zoloft. They work the same way to block the reuptake transporter for serotonin, but they are different chemicals and sold by different pharmaceutical companies. But they all work in the same way. Serotonin, once released from the terminal, can be found in the synaptic cleft. It is removed from the synaptic cleft through a transporter specific for serotonin, and these antidepressants or SSRIs once they activate receptors, but these SSRIs block the transporter, right? Block the transporter, keeping serotonin in the synaptic cleft longer, allowing it to continue to activate its receptor on the postsynaptic cell. And that seems to help some people who suffer from depression. What's crazy about serotonin, and this is unlike any other kind of neurotransmitter, is that to date, there are at least 17 different subtypes of serotonin receptors, which is insane. I should also mention, because serotonin is a monoamine, once it gets into the terminal, it's also broken down by MAO, monoamine oxidase. On to now acetylcholine. Acetylcholine is different from other neurotransmitters that we've discussed so far, so it kind of gets a category on its own. I'm going to be abbreviating acetylcholine as ACH. The synthesis of acetylcholine, because it's a classic neurotransmitter, also occurs in the terminal button. Acetylcholine is made from choline, which we get from our diet, and a byproduct of metabolism called acetyl coenzyme A. The enzyme choline acetyltransferase, so even though acetyl coenzyme A has the word enzyme in it, it's not an enzyme. Choline acetyltransferase is an enzyme. It combines the acetyl from acetyl coenzyme A and choline to make acetylcholine, the active neurotransmitter. You can abbreviate choline acetyltransferase as CHAT, right? Because it's a lot easier to say than choline acetyltransferase. But choline acetyltransferase is the enzyme responsible for taking the two precursors of acetylcholine and making the active neurotransmitter acetylcholine from it. From here, it gets packaged into vesicles and released through exocytosis. It can activate receptors in the postsynaptic cell, and we'll talk about its receptors in the coming slides. But here's what's unique about acetylcholine compared to monoamines and indolamines. Catecholamines and indolamines. Acetylcholine is broken down by enzymes in the synaptic cleft. The enzyme responsible for breaking down acetylcholine is called acetylcholinesterase, or ACHE, right? We like abbreviations instead of using the long words. So ACHE is an enzyme that takes acetylcholine and breaks it down into choline and acetate. The choline then gets pumped back into the presynaptic terminal to be reused, right? Why would we waste it? To be reused to make more acetylcholine, and then acetate diffuses out and kind of enters the bloodstream. So in this way, rather than waiting for the next round of choline from our diet, we can reuse, our nervous systems reuse the choline that gets broken down from the active neurotransmitter acetylcholine. And so your textbook kind of gets at the synthesis of acetylcholine the same way. Acetylcoenzyme A, which comes from breaking down substances, and choline, which could come from metabolism but also comes from diet, is used to make acetylcholine, the neurotransmitter. There are two major receptors for acetylcholine. They are nicotinic receptors and muscarinic receptors. Nicotinic receptors are ionotropic. Muscarinic receptors are metabotropic. Nicotinic receptors got their name because the chemical nicotine, which can be found in tobacco products, activates these receptors to produce their effects. And there is a chemical found in a mushroom called muscarine, which activates muscarinic receptors. But nicotinic receptors are ionotropic. They're ion channels, whereas muscarinic receptors are G protein coupled receptors. All voluntary muscle movements in your body occur through the actions of acetylcholine acting on nicotinic receptors specifically. We want quick, fast reactions to things that happen in our body, and so all the skeletal muscles in your body utilize nicotinic receptors and acetylcholine to produce contraction. You'll also find acetylcholine in the autonomic nervous system, but it plays a role in REM sleep, as well as learning and memory, and is thought to be involved in dementia like Alzheimer's disease. The last category of neurotransmitters that we're going to be talking about today are neuropeptides, these large neurotransmitters. And you may be familiar with or have heard of endorphins. Endorphins are a type of neuropeptides, but there's a ton of them. You may have heard of oxytocin or corticotropin releasing factor. These are all neuropeptides, and there's hundreds of them. So we're not really going to spend much time talking about them because this category of neurotransmitters is so big. But like all peptides or proteins, neuropeptides are synthesized in the soma using dRNA to make mRNA. Then a precursor protein is made, and enzymes break that precursor protein down into the peptide or active neurotransmitter. The peptides get packaged into vesicles in the Golgi apparatus in the cell bodies, and then transported down the axon to the terminal. Some examples of neuropeptides that you may have heard of that I've mentioned already: endorphins. These are types of opiates, but they are made by your body. So they're similar in action and structure to morphine or heroin, but they are not as powerful as those drugs. But they activate the same receptors as those drugs do, endorphins do. Oxytocin is a neuropeptide that's important in birth and lactation, and is also thought to play a role in kind of social bonding, in addition to its function in birth and lactation. But neuropeptides have a variety of different functions in the nervous system, and the vast majority of peptide receptors are metabotropic. Unlike classic neurotransmitters, which can be broken down by either reuptake or enzyme degradation, peptides are almost always broken down by enzyme degradation. So let's talk about something that many people are really interested in, which is how do drugs affect neurotransmitter function to produce the effects that they do in the nervous system. So their drug action depends on a variety of different factors, one of which is how the drug is administered, how quickly it gets absorbed and distributed throughout the brain. Does it have sites in the body where it can be stored, where it's not doing anything but just kind of hanging out? Sites like fat and bone. How quickly does it get broken down? How does it get excreted? these 6 factors influence how well a drug can bind to its target site to produce its effect. So for example, if it takes a while for the drug to get into the bloodstream and the brain, the effects are not going to be as powerful as drugs that can quickly get into the bloodstream and to its target site, like inhalation or injection. But again, this is something that we'll talk about in more detail in a course called psychopharmacology, which is something that I teach as a capstone in the program. The other important piece when we're talking about drug use and drug action is something called therapeutic versus toxic effects. So we want to make sure that the drug is producing. So drug efficacy talks about how effective the drug is at producing the outcome or effect that the drug is designed to do. But all drugs have side effects, actions that are unintended, but the drug still produces. And so the drug may require a certain level of dosage in order to produce the therapeutic effects, like the effect we desire it to produce. And it might also produce other effects that we don't want it to. So these are called side effects. This particular chart is using morphine as an example to illustrate the drug's effectiveness. In order to produce pain relief, right, most drugs, when we increase the dose, produce this S-shaped curve. So this S-shaped curve is what we see for pain relief, which is often what morphine is used for. But too much morphine can actually kill you, and it can produce difficulty breathing, right? Which may slow down your breathing to the point where it kills you. The safe dose is this range of dose where we get more therapeutic effects and less dangerous side effects. And so typically when we administer drugs like morphine or any other drug for that matter, there's a range of doses that are used to administer the drug where it produces therapeutic effects, but it minimizes the effects of toxic side effects. Okay, so that's called therapeutic range of drugs. So how do drugs produce their effects, right? We've talked about a general feature of neurotransmitter, of a neuron. It's got a cell body, dendrites coming off of that cell body, as well as a long axon where action potentials occur. And then once that action potential gets to the terminal, it releases neurotransmitters. So the vast majority of drug action happens at the synapse. So we're going to be hanging out here talking about the different ways in which drugs produce their effects. But before we do that, I want to introduce you to different kinds of receptors. So far we've talked about postsynaptic receptors, receptors that are found on the postsynaptic cell and produce an effect on the receiving neuron. I want to introduce you to a type of receptor known as the autoreceptor. Autoreceptors can actually be found in the presynaptic, where's my cursor? Presynaptic neuron, right? Presynaptic membrane. And autoreceptors bind to the neuron's own neurotransmitter. So whatever neurotransmitter is being released, it also can bind to autoreceptors. Autoreceptors have a really important function. A neuron has no idea how much neurotransmitter it has released, right? It can continue releasing neurotransmitters without having any clue as to how much neurotransmitter it's released. There's no feedback. Autoreceptors serve as the feedback for neurotransmitters. So almost all of the neurotransmitters we've talked about, the classic ones in particular, have autoreceptors that allow a neuron to monitor how much neurotransmitter is being released. So when an autoreceptor, which is found on the presynaptic terminal, gets activated by its own neurotransmitter, it triggers the decrease in production and release of that neurotransmitter. So it is kind of the monitor or gateway for that neuron to know how much neurotransmitter is being released. So when autoreceptors get released, it tells the neurotransmitter, oh, you've been releasing too much. It's time to either slow down release or slow down production. You can have drugs change the release of neurotransmitters by activating or blocking autoreceptors. And we'll talk about that in the coming slides. So don't worry about the green drug here. We'll talk more about it in a second. But I wanted to introduce what our autoreceptors were in this particular slide. Okay, so the locations or the places where drugs can affect neurotransmitter function, it can do that in these 3 steps that I'm going to be introducing you to, which you should know already, but we're putting it together in one complete slide. So here is our terminal button, our presynaptic terminal. Here's our postsynaptic cell. The very first step of neurotransmitter action is making the neurotransmitter. So precursors are typically used to make the neurotransmitter, and enzymes are the ones that convert the precursor to the active neurotransmitter. The second step is storage. Neurotransmitters need to be stored into vesicles in order for them to be released. The third step is the process of exocytosis. There are multiple steps involved in exocytosis. The fourth step involves the neurotransmitter binding to its postsynaptic receptor on the receiving cell. The fifth step is the activation of the autoreceptor. If the autoreceptor is activated, it will stop neurotransmitter release. The sixth step is the removal of the neurotransmitter from the synaptic cleft. And there's 2 ways we've discussed that neurotransmitters can be removed, either reuptake through the transporter or enzyme degradation. So these represent the 6 steps in neurotransmitter action, and a drug can affect any 6 of these steps to modify neurotransmitter function. So let's talk about the ways in which drugs can modify neurotransmitter function. There are 2 major ways that a drug can affect neurotransmitter function. The first is that it can increase neurotransmitter action, in which case the drug is known as an agonist. So if it increases or facilitates what the neurotransmitter would normally be doing, that drug is called an agonist. If a drug decreases neurotransmitter action, so it prevents the neurotransmitter from doing what it normally would want to do, it's called an antagonist. And these drugs can act at any 6 of those steps in the prior slides of synaptic transmission to produce agonistic or antagonistic effects. Okay, so a reminder, agonistic effects, we're increasing what the neurotransmitter would be doing. Antagonists decrease the actions of that neurotransmitter. All right, let's talk about what drugs can do at each of these steps, right? So I'm going to give you scenarios, and before you move on, I want you to pause the video and take a guess as to what that drug would be. Would that drug be an agonist or an antagonist based on its effects? So the first one, if a drug were to increase or serve as the precursor for the neurotransmitter, would it be an agonist or antagonist? Let's do this together. If a neuron has more of the precursor, it's going to make more of the neurotransmitter, therefore increasing neurotransmitter action. And so this scenario, the drug is an agonist. If we destroyed enzymes or blocked enzymes that make the neurotransmitter, I'd like you to pause and take a guess, we would be decreasing neurotransmitter action because we aren't able to make more neurotransmitters, and therefore that drug would be an antagonist. if we prevented storage of the neurotransmitter in vesicles, which is the only way the neurotransmitter can be released, the drug would be an antagonist. If we stimulated the release of the neurotransmitter by enhancing exocytosis, more neurotransmitter would be released, and therefore that drug would be an agonist. If we decreased neurotransmitter release by blocking, let's say, voltage-gated calcium channels, right, blocking exocytosis, we would decrease neurotransmitter action. The drug would be an antagonist. If we stimulated or activated postsynaptic receptors with a drug, that drug would be an agonist. Conversely, if we blocked postsynaptic receptors, preventing the neurotransmitter from activating the receptor, the drug would be an antagonist. If we stimulated the autoreceptor, this is important, this is a little bit opposite of what you might expect. Activation of the autoreceptor is going to reduce neurotransmitter release. So if we stimulate the autoreceptor, we're decreasing neurotransmitter release, therefore decreasing neurotransmitter action, and the drug would be an antagonist. If the drug blocked the autoreceptor, that means the neuron is not getting the message that it's releasing too much neurotransmitter, it would continue releasing neurotransmitters. We would get increased neurotransmitter action, and the drug would be an agonist. If we block reuptake, which is one of the two ways we can remove neurotransmitters from the synaptic cleft, the drug would be an agonist because the neurotransmitter would hang out in the synaptic cleft longer. If we block degrading enzymes, the same thing would happen. We would increase neurotransmitter action, keeping the neurotransmitter in the synapse longer, and therefore the drug would be considered an agonist. So if this was a little bit hard to understand, I would go back and rewatch and try to make sense of what is happening if we create a drug that does each of these scenarios that I described to explain why it is an agonist or antagonist. So it's really important that you understand what each of these steps is doing so that you can understand why actions at these 6 steps can either increase or decrease action of the neurotransmitter, making the drug an agonist or antagonist. Your textbook has a diagram here that kind of summarizes similar actions. The only one that we haven't talked about is retrograde transmission. Don't worry about that. You won't be responsible for it. But we've talked about more of these than your textbook illustrates. So I do want to mention, so one of the things that we didn't talk about in that prior diagram is increasing or decreasing action potential. That's going to dictate whether the drug is an agonist or antagonist. So if a drug increases action potentials or rate of firing, the drug would be considered an agonist. If we decreased action potentials, the drug would be an antagonist. And I'd like you to think about why that is. All right, so now let's talk about drugs themselves, right? I mentioned being addicted to caffeine and loving caffeine to start my morning off, and you may be one of those individuals too. How exactly does caffeine work in the brain to produce increases in arousal and attention? So we haven't talked about the neurotransmitter adenosine, but adenosine is an inhibitory neurotransmitter that acts in the brain. Adenosine binds to what are called adenosine receptors, and it produces inhibitory action. So very similar to GABA, but it's a different kind of neurotransmitter. So when adenosine binds to its receptor, the neuron is less active, contributing to decreased arousal, attention, and increased sleepiness and tiredness. What caffeine does is it blocks the adenosine receptor, preventing adenosine from binding to it. When adenosine is not able to bind to its receptor, the receptor is blocked. Okay, so adenosine caffeine is an antagonist for adenosine, but because adenosine can't decrease neuronal function, neuronal activation, we become more aroused, alert, and awake, right? So caffeine blocks the actions of adenosine, which is to make us sleepy and tired and drowsy. And because it's blocking adenosine receptors from doing what they can do, caffeine is an antagonist for adenosine, but its effects are to increase arousal, alertness, and awareness. So if you're a caffeine addict like me, you now know how caffeine works to keep you more awake. There are other drugs that you might be familiar with, like cocaine, for example. And again, if you're really interested in this topic, we can spend some time discussing it in psychopharmacology, but here's another example of how a drug can produce its effect. Cocaine is a drug that modifies dopamine function in the nervous system. What cocaine does is it blocks the reuptake of dopamine, so it's blocking the reuptake transporter, causing dopamine to hang out in the synapse longer and ultimately producing increased response of the postsynaptic neuron. So cocaine is an agonist for dopamine because it's enhancing dopamine function by blocking its reuptake. There are several different types of psychoactive drugs. We have large categories of psychoactive drugs listed here, and their general functions as well as examples of drugs that fall into that category. We have CNS stimulants, the ones that kind of activate the nervous system and generate action potentials. We have CNS depressants, the ones that kind of block action potentials and activity. We have drugs that decrease pain, analgesics, drugs that change our perceptual experience, hallucinogens. There are other drugs that produce varied effects in the central nervous system, so they don't necessarily fit into one of these categories above. And finally, we have drugs that serve as therapeutics to relieve disorders of the nervous system. So, drugs like amphetamine, cocaine, caffeine, nicotine are all considered CNS stimulants. They activate, arouse the nervous system. Alcohol, barbiturates, benzodiazepines are all called CNS depressants because they suppress CNS activity, promoting relaxation. Pain relief can be modified by drugs like opiates, morphine, codeine, heroin. We can modify perceptual experience using hallucinogens like mescaline, LSD, or psilocybin. Drugs like marijuana don't neatly fit into any of these upper categories because they produce a whole bunch of different responses. And so it gets a category on its own, but it can be used both for medicinal purposes, but it can also produce psychoactive symptoms, including mild hallucinations if given at larger doses, but it can also promote relaxation at lower doses. Psychotherapeutics are drugs that are used to treat disorders, so drugs like Prozac or Thorazine, an antipsychotic, falls into this category. So that's it for this particular lecture. We've talked about a whole bunch of things in this particular lecture. We discussed the different types of neurotransmitters, the categories of neurotransmitters, of classic as well as neuropeptide neurotransmitters, mentioning the different types of receptors each has and how those receptors and neurotransmitters control varied functions in the nervous system. We talked about the 6 steps of neurotransmitter action and how these drugs can modify nervous system function by acting at any of these 6 steps. Depending on the drug's effects, it can be an agonist if it promotes neurotransmitter action, or it can be an antagonist if it decreases neurotransmitter action. And there are several categories of psychoactive drugs, drugs that act in the nervous system and produce psychological or behavioral changes. Some increase it while others decrease it depending on the drug. So that's it.